Malaria is an infection by a parasite that is particularly destructive of human populations that live in warm climates. The reason that this is the case is because the parasite's life cycle is reliant on transmission by mosquitoes, which are most commonly found in warm equatorial climates. The parasitic infection of the blood leads to the disease malaria, which occurs after the parasite begins feeding on and utilizing human blood cells to carry out its reproductive cycle. Essentially, you can imagine it as the parasite eating human blood cells. Malaria occurs after the degradation of these cells leads to the buildup of toxic byproducts in the bloodstream. The buildup of these products leads to symptoms in humans of fever, chills, sweats, headaches. The disease can oftentimes be fatal, particularly so since treatment options are limited in many of the regions with the highest incidence of this disease. In the most serious cases, the damage to the infected erythrocytes can aggregate in the brain and lead to cerebral malaria, which is the form of the disease that has the highest incidence of death. The study that we observed discussed a possible mechanism explaining how sickle cell disease may provide resistance to malaria. Scientists have observed increased survivability in human populations within those who have sickle cell disease and who have been infected by malaria. So the question postulated by many is, why is this the case? Sickle cell disease occurs by way of genetic mutation. In this disease, the human hemoglobin has exchanged one amino acid at the 6th position for another, which leads to misfolding of the hemoglobin protein. This misfolded protein, when deoxygenated, can structure itself into a sickle shape and aggregate within the vasculature, leading to decreased life expectancy for the individual with the disease, anemia, and increased risk of blood clotting. It is observed that sickle cell disease is more prevalent in equatorial populations, and some have theorized that this disease has co-evolved alongside malaria because the negative survival advantage of having sickle cell disease is counteracted by the positive survival advantage of being resistant to malaria.
It had been indicated in previous research that humans with point mutations in the beta chain of human hemoglobin have a resistance to Plasmodium infection. This occurs in humans who are carrying the mutation in the heterozygous form. Humans who have the mutation in the homozygous form will present with sickle cell disease. It was also indicated in previous research that individuals who have the heterozygous form of the HBS mutation will accumulate low levels of heme in their plasma. This in turn triggers the expression of heme oxygenase 1. This enzyme catalyzes free heme into biliverdin, iron, and carbon monoxide. The expression of heme hydrogenase has previously been shown to have a protective effect on many immune-mediated disease. Most relevant to this study is a protective effect against experimental cerebral malaria in mice, a disease in mice which mimics some of the pathological features of cerebral malaria in humans. It was found that this protection is mediated by carbon monoxide. The carbon monoxide, in a sense, binds to the free hemoglobin and inhibits its oxidation, which prevents the release of heme. The researchers had hypothesized that the expression of heme oxygenase and the production of carbon monoxide in individuals with an HBS mutation would explain the survival advantage that is experienced by those with sickle cell trait. They sought out to identify the specific molecular mechanism which is responsible. The researchers confirmed the hypothesis and found that the induction of heme oxygenase 1 by sickle hemoglobin involves the NRF2, a transcription factor. For the experiment, the researchers used HBSAD mice, which are mice with a hemoglobin mutation, which makes them a suitable analog for human subjects. The results showed, interestingly, that the protective effect occurs regardless of parasite load and parasite sequestration in different organs. The researchers concluded that it was the NRF2 heme oxygenase 1 system which was the specific pathway that they were looking for. It was particularly interesting that the researchers found that a certain level of heme in plasma would have a protective effect because it was broken down by heme oxygenase and produced carbon monoxide. But they also found that levels of heme in the plasma which were too high had a significant cytotoxic effect and added to the pathogenicity of the Plasmodium infection. One of the main limitations that we noticed in this study was that the experiment was done on mice, specifically mice with hemoglobin SAD. Although it is widely accepted that experiments on these types of mice are a suitable analog for human subjects. The study was extremely scientifically important because by indicating this specific pathway and mechanism which affords individuals with heterozygous hemoglobin mutation, it opens the door for treatment of the infection and possibly prevention or a cure. With this in mind, further research could be done into whether it be beneficial to induce heme oxygenase in mice with normal hemoglobin to see if it would impart the same protection experienced by mice with sickle hemoglobin. This could be done by slightly elevating the plasma heme levels. There could also be research done which focused more on raising carbon monoxide levels in mice with sickle cell trait to see if the treatment is suitable enough to have a protective effect against Plasmodium infection.
This study, of course, focuses more on the how sickle cell offers a resistance to Plasmodium or malaria infection, rather than the why is this happening in the first place. This aspect, the relationship between mutation and potential cause of said mutation, is actually the most interesting topic that I personally feel arises from this discussion. Are we in fact witnessing evidence here of modern-day human evolution? This concept, of course, of environment favoring certain mutations over others is nothing new, even in modern times. For example, we see how living at extreme altitudes has caused the Nepalese Sherpas to change their mitochondria to more efficiently use very limited oxygen to produce ATP. We've also seen how peoples of Northern European descent, a land of very cold and brutal winters, developed a mutation about 3,000 years ago that allowed them to digest dairy products into adulthood, a trait formerly shut down after weaning off of their mother's breast milk. This allowed a great many people to build up fat stores from the abundant, calorie-rich dairy products, which helped them get through the bleakest winters. I think the real significance in this sickle cell malaria case, however, is that it's not just an environment which favors a particular mutation, but rather an environment which hosts a vector, in this case a mosquito, which hosts a parasite. We're now beginning to see that very specific and perhaps even tailored changes in the world we live in can and does lead to evolution. Though there are those who have stated that humans have evolved as far as we naturally can go, relying now only on our minds and scientific inventions to further our species, this is clearly not the case. With the addition of antimicrobial-resistant bacteria coming onto the scene, a result from all of our heavy antibiotic usage, the need for mutation and evolution to keep us healthy begins to trump the call for science and medicine. I'm sure most would agree that the human race is far from perfect, though each evolution does try to bring us one step closer.